Warm welcome, good morning everyone again, and especially to all the participants who will arrive a little bit later. With me, we start the masterclass, the first masterclass of today with the topic cancer treatments and organ generation, novel approaches to soft tissue. And with me here is Professor Kurreck, Julien Payen and Philippe Marchetti, and please have a seat here. And our fourth speaker is unfortunately ill. He's not with us, and for him we wish all the best and a quick recovery. And you have a little bit more time right now for your presentations and for the discussion later on. And let's start with Professor Kurreck. He's the Professor of Applied Biochemistry and Director of the Institute of Biotechnology at the Technische University here in Berlin, and his research focuses on the development of human three-dimensional organ models using bioprinting, which can then be used for infection or cancer studies as an alternative to animal testings. And yes, I would say the stage is yours.
Okay, so thank you for the kind introduction and the opportunity to present our results. I will switch gears a little bit. Up to now, we have mainly talked about very solid materials. Now I will talk about soft materials like organs and introduce our project on the bioprinting of cancer models. So our background is that we want to somehow improve or contribute to the standard drug development, and the standard procedure is that we usually work in 2D cell cultures and then move on to the animal model. The 2D culture has the disadvantage that, of course, there is no three-dimensional interaction between the different cell types that we have in a biological organ. And the animal model has the disadvantage that there is a limited predictability of the results obtained from animal models for the human being. And in addition, of course, there is an ethical pressure that we should reduce the number of animal testings. And but the main problem we see is that 97% of all cancer drugs that have undergone preclinical testing fail during the 3 clinical phases of testing. And this is mainly due to the species-specific differences between mice and men. So we think that 3D bioprinting offers an alternative because here we can provide pure human models. Obviously, this is an audience that is familiar with additive manufacturing, so I don't have to introduce the 3D printing technologies. The only point I want to make is that according to the definition we use, bioprinting means that living cells are present in the process of in the printing process. In a broader sense, bioprinting can also include printing of scaffolds that become populated with cells afterwards, but in the strict sense, the cells are already present during the printing process. We all know that there are dozens of 3D printing technologies out there. The main technologies used in bioprinting are summarized here, and we mostly use pneumatic and mechanical microextrusion. We use a little bit of inkjetting and stereolithography, or to be more precise, digital light processing. So what is also important to know is that there is no very best optimal technology for bioprinting. It's always a question on what you're aiming at. So we need different types of printers to cover the different technologies we need for bioprinting. So here you see our printer park. So we have printers for digital light processing. We have printers for mechanic and pneumatic microextrusion. And of course, we have some printers like conventional FDM and stereolithography printers that we use to produce small parts for our processes. And my favorite printer is in the middle of the top row. It's a chocolate printer, so that is for the leisure time in between. So the first short story I want to tell you is a very recent story. We noticed that the second component is also important. It's not only the printer, but it's also the bioink. As I said, the bioink usually comprises living cells and a hydrogel. And so we did a competition, man versus machine, and a US friend of mine said that reminded him of an old US folk hero, John Henry, who drilled holes for railroad tunnel construction and took a competition against a steam-powered drilling machine. And he won, but he was so exhausted that he died. Converting this story from 1870 to nowadays, as I expected, the movie doesn't run on this system. The conventional bioink mixing that everybody uses around the world is that you fill the hydrogel and the cells into two syringes. And now it works. Here is the first syringe where you have the first component, and this is the cell solution, and then you add the hydrogel, and then you mix manually. That's what everybody does around the world. And we now developed an automated mixing machine where you can also fill the two components and then mix them. And then we did a challenge between man and machine. So my PhD student asked 3 lab members to do the manual mixing. He never told me who is who, but what you see is that the 3 different operators had 3 completely different outcomes with regard to cell distribution, with regard to cell viability. Even after training all the operators, there were still large deviations. And I think that this is a big issue when comparing results from different groups around the world. So then he, as the most advanced operator, competed with the machine. And still the machine won. As you can see in the lower panel, that there is a very even distribution of cells and a very high cell viability. So as I stressed several times now, the importance of the cells and their viability, and that can be substantially improved with this mixing machine as compared to the conventional mixing. So if we have a good bioink distribution, the next thing is what do we want to do? We want to produce organ models. And there are different technologies out there to do 3-dimensional cell culture. There are organoids, different types of 3D cell cultures, but most of them cannot include a vascular system into the 3-dimensional organ models. And this is the main advantage of bioprinting. And so here you see several constructs where we included a vascular channel to our printed organ models. And in our first story, we printed a lung cancer model, and we had a vascular channel in this lung cancer model, and then we connected this to it was produced by digital light processing, and we connected the model to a peristaltic pump and pumped the media through this model. And then, so here you see the design of the model and the printed model. Here you see the real setup. You have the peristaltic pump, which pumps the media through the model. And on the lower part of the middle figure, you see blue ink flushing through the model. And it actually is perfused through the channel, but also diffuses into the model. Of course, this is what we want. We want to supply nutrients and drugs for testing in the model. And what we found is that we have a substantially and significantly increased cell viability on the dynamic cultivation with the perfusion system as compared to a static cultivation, which is the standard way. And we then tested various cancer drugs and found that they are highly active if we apply this perfusion. And then we further improved this system and developed a method of sacrificial-free direct ink writing. So the point is that there are many approaches out there on introducing these vascular channels into bioprinted organ models, but usually they use sacrificial materials like pluronics that can be removed after the printing process. But my student, Dongwei Wu, he developed a method he optimized the bioink and the printing conditions to directly include these vascular channels into the organ models without the need of a sacrificial material. And the blue part in the center is now a cancerous part, and the surrounding part is a healthy tissue, for example, liver tissue. So what we simulate here is the formation of metastasis in the liver. And then he produced different designs, like a single channel, a branch channel, or a more natural network. And he connected this to a perfusion system again. In this case, he used micropumps that he can include in the multivalve plate and introduce into the incubator. And another important part that he did was that he used agar casting to, first of all, seal the connection between the tubings and the model, and secondly, to improve the stability of the model. So with this, we can carry out long-term cultivation of the model. And then another important part is that blood vessels are lined by endothelial cells, and we also simulated this. So after the printing process, we added endothelial cells to our models, and it was important to rotate the model because on the top right, you see on the left part something that was just where the endothelial cells were just added, so they only line in the lower part of the vessels. But if you rotate the model, then you get a full lining, full population of the vessels with endothelial cells. And finally, the actual question was, can we use anti-cancer drugs, cytostatic drugs in our models? And he used a prodrug that is an inactive form of the drug that will not inhibit cancer growth. He had MCF7 cells, this is a breast cancer cell line, as a tumor model surrounded by hepatic cells. And the important part was that the hepatic cells, after activating of cytochrome P450 enzymes, were even capable of activating this inactive prodrug to its active form to have the cytostatic activity on the cancer. And on the lower right, you see that only with the activation of the metabolizing enzymes and the prodrug, you get a reduction in cancer spheroid growth. So what we think is that we now have a nice model. It's purely composed of human cells, so it's human cancer cells in a human microenvironment, which differs from the typical mouse model where we implant the human cancer into a murine microenvironment, and we can use this model for further testing of new cancer drugs, and we are in collaboration with the Charité to test their substances they are currently developing. And with this, I want to thank my group, the collaborators, and the funding agencies. And thank you for your attention.
So, thanks Jens Kurreck for this fantastic presentation. I love all these bioprinting topics. And before we go further on, please keep your question in mind or send it to the Slido system. And after the next presentation, we will discuss all your questions together. Now I would like to hand over to Philippe Marchetti and Julien Payen. Philippe is Professor of Cell Biology at the Faculty of Medicine, University of Lille. And he has more than 20 years experience in INSERM laboratories of cancer research and specialized in the study of cell death and its control by mitochondrial metabolism. And Julien, he's the CEO and co-founder of Lattice Medical. Some of you maybe knows him from the 3D Pioneers Challenge. This year he's the winner of this year's challenge, and he started his career in respiratory medical devices and FFP2 face masks at Mako Pharma and Honeywell. And from that, he met Dr. Marchetti and his colleagues in Lille Hospital and decided to create Lattice Medical to disrupt breast reconstruction. So the stage is yours. I'm very curious.
Thank you.
Thanks.
So thank you for the kind introduction, and ladies and gentlemen, nice to meet you. So Lattice Medical is developing advanced solutions for the soft tissue and reconstructive surgery. And we try to merge biomaterials, 3D printing, and tissue engineering to do that.
Okay. So, as you may know, breast cancer is the most common cancer in women. It affects one woman out of 8 worldwide, and the major treatment is surgical ablation, also called mastectomy, and approximately 240,000 patients undergo mastectomy each year. Unfortunately, only 20 to 30% of those patients will benefit from breast reconstruction, and this is mainly due to the absence of fully satisfactory solutions. The current breast reconstruction solutions are first silicone implants, but everybody knows the potential complications with those implants, like lymphoma. Alternatively, autologous surgeries such as lipofilling or flaps have been developed. Those procedures are very efficient, but they are often complex and highly invasive. So there's an urgent need to find new solutions. Moreover, women are looking for natural, minimally invasive, and long-lasting breast reconstruction. That's why we introduce a breast revolution with the Lattice bioprosthesis. This novel implant allows regeneration of autologous fat tissue. This implant is degradable, avoiding long-term complications, and it can be placed, and it's really an advantage, in a simple and single surgery of only 1 hour.
So I'm sorry for the video next before the lunch. We try to explain the operative technique surgery on this example. So the patient here had mastectomy, a full mastectomy on the left breast, and the entryway is in the mammary fold. So thanks to a Doppler echography the day before the surgery, we will seek for a fat tissue. This fat tissue is located between the 7th and 8th intercostal area, just here, below the breast. And the surgeon will harvest 80 to 100 cc of fat from here and put the fat inside the implant, Lattice. So on the scaffold, it fixed the tissue with 6 stitches and closed with the chamber. The chamber can have different size and shape thanks to 3D printing. Once it's prepectoral fixed, we close the wound, and during 3 to 6 months, the tissue regenerates inside, fills completely the inner part, and in the meantime, the implant disappears. After all, the patient gets back a new breast with its own tissue without any device. We have incorporated Lattice Medical 6 years ago in the north of France. We raised the company with Philippe and 2 other colleagues, medical doctors and plastic surgeons. And we are also followed with medical experts in the field of plastic surgery in France and US. We are 25, and like every startups, we continuously raise funds for the research.
Okay. Before clinical trials, we have carried out over 10 years of preclinical research, and here is a summary of performance of tissue regeneration in 10 mini pigs. According to MRI images, you can see here the fat flap grows, the fat flap is in white, grows over time. And it's under the Lattice implant represented by black dots. You have the image at day 90 post-operative, where the regenerated tissue is present totally inside the implant. When the volumes are calculated, it's the right panel. When the volumes are calculated, you can see the progressive growth of the fat flap over 6 to 9 months. Then it fills almost completely the entire implant, exactly 99%, but it's also important to note that the fat flap volume is stable over a 21-month period. So to go further and to have a better characterization of the regenerated tissue, we performed histological analysis. As you can see here, the fat flap consists of a viable mature adipose tissue without necrosis or signs of inflammation, and this normal tissue is surrounded by a thin layer of connective tissue. The extensive tissue ingrowth is supported by strong neovascularization, as illustrated by CD31 positive blood vessels here in green. And macroscopically, you can see the blood on the surface of the fat flap. Finally, we check the absence of systemic inflammation, and there's no change in serum protein levels over time. Finally, NMR analysis did not show any accumulation of degradation products of the implant in kidneys and liver after 12 months of implantation.
So following these preclinical results, we have been granted this year to start a first-in-human clinical trial on women undergoing breast reconstruction after a total mastectomy. Two kinds of patients can be included in the study. Patients with immediate reconstruction, so we can operate both mastectomy and reconstruction with Lattice in the same surgery, or a deferred reconstruction. So it's a first-in-human, so safety and performance study are the primary endpoints, and it's a 3 years follow-up, and we will include 50 patients until June 2025 in 8 centers in France and Spain. And we work with several key opinion leaders in Europe which are leading this study. For instance, we've got some results on first patients. What we can say first, is the surgical time. Instead of 4 to 8 hours of flap surgery, we can reduce the surgery time of only 1 hour and perform in the same surgery at 1 hour and a half for mastectomy and reconstruction. It's not painful for the patient in 1 on 10 on the pain scale, and we can show that we have a regrowth of tissue at only 3 months. On that case, the first patient here, 63 years old, on the right breast, she got a full mastectomy, and she get back with, thanks to Lattice, a new breast in only 3 months. And like preclinical results, we use MRI to check the volume regeneration, and the volume regenerated by 2 in only 3 months. So it's first positive outcomes, and maybe next year we can show you more results and consolidated results on several patients after 1 year of review. We have also raised our own manufacturing tool, a 3D printing production platform. So we are able to extrude the biopolymer on the left to make the filament. You see, we use really common standard printers. This printing farm is compliant to ISO medical standards. We run in ISO 7 clean room for the environment, and we can produce, for the moment, 1,000 implants per year, thanks to this printing farm, 16 printers. we have some kind of post-processing, washing of the device, and then we have the quality control inspection. I was really interested on the last presentation before on the visual inspection. It's really interesting to have it online. And the device is packaged in a double blister and then sterilized to be ready to be implanted. So we also made the proof of concept of manufacturing with 3D printing. We are not yet on custom shape. We want to demonstrate the feasibility on high volume production with 3D printing, and it's totally scalable and flexible. And thanks to this, we can develop several other applications in the soft tissue. So you see Lattice for the breast reconstruction. We have the other one device at preclinical level. It's Rodin for the skin regeneration after severe burns, after deep wound. So based on the same idea, you inject fat into the device, and then the fat colonize and the device disappear. And the last product now released is a mesh reinforcement tissue for reconstruction. And we have also developed medical filament for 3D printing, and it's branded to Lattice Services, a commercial brand of our filament. And we have two kinds of filaments: absorbable filament, medical grade, with different kind of polymers, PLGA, which is strong and dedicated to bone applications. PDO and PLCL more dedicated on soft tissue applications with different resorption rates, and the caprolactone also. And we for the moment work with universities in the development of several medical devices. And we have also a non-implantable material, so medical grade skin contact filament with ABS polycarbonate, which is can be sterilized in autoclave, for instance, a soft filament and PETG. And you understand, we have the raw materials, we have the production platform, so we can help our customers to develop, to design 3D printed products and test with our lab different in vitro or in vivo tests, and then pass to the production and test at clinical level. So we have a booth. Florian, our business developer, is here, and feel free to ask any questions. Thank you. Thank you very much.
Thanks a lot. Thanks a lot to you both guys, and I'm sure, and I see there are lots of questions, but before we start, I have also two questions. First addressed to Jens Kurreck, because since I'm only coming from the tissue engineering sector, I'm highly interested in how long can the printed organ models be cultivated, because there are lots of risks for contamination during the cultivation time. So what's your point of view?
Actually, this is one of the great advantages of the vascularized model systems. So we did cultivations up to, I think we had one model for 35 days, but there was no drop in cell viability, so we could have done longer, but we simply stopped at that point. Of course, sterility is a big issue then. Let's be open, in an academic environment where you have a lot of students, it's a bit challenging. But as long as we keep the sterility, the models can be cultured for a rather long time compared to a conventional 2D cell culture.
Okay. And is it an advantage to use the machine instead of manpower?
We have not directly compared this, but as the cell viability initially is better and the cell distribution is better, I guess it's better.
Mm-hmm. Thanks. And one question to you both. It's a personal question because I'm a woman. How comfortable is it to wear such an implant? It looks not so flexible, especially in the first days or weeks. It looks like a little bit stiff if you printed it. It's an FDM process. How comfortable is it to wear it?
Answer? Okay. So yeah, this is one of the questions we assess starting the clinical trials. And yeah, it's softer at 37 degrees. The TG of the material is softer at the body temperature. And based on the pain scale of the patient, she come back the first month, 3 months, 6 months on the protocol. So they did not feel really hard and tough. And thanks to the resorption, after 4 to 6 months, it gets really, really softer, and the patient don't feel it after 6 months.
Okay.
It's soft outside the body, but soft inside.
Okay. Okay.
Can I ask a question?
Yes, of course.
Yeah. Thank you for your nice presentation. I would like to know how many cells you can mix together. What is the maximum number of different type of cells?
Different type of cells.
Yes.
I don't think we have a limit. So currently our most complex model is a lung model, where we have 3 different layers with 4 different cell types. So we have an epithelial layer, a fibroblast plus immune cell layer, and an endothelial layer. So these bioprinters can accommodate different print heads. So we can, the Bio X6 has 6 print heads, so we could combine 6 different bio inks, each containing different cell types.
Okay, so there's no limitation? Theoretically?
Yeah, I don't think so. Yeah, right.
And maybe the limitation is the cell culture medium later on.
Yes.
That is a major issue. If you do the co-cultures, we see that the cells communicate and it's advantages, but each cell has its optimal medium, and this is the big challenge to find a cell culture medium. It sounds like a technical issue, but it is the most important part, to find a good medium that is suitable for all cell types you want to combine.
And bio-ink is also a technical
Bio-ink is not so much of a problem. No? Most of our bio-inks are based on alginate gelatin mixtures, which is the common procedure. And this is pretty good for all cell types we have tested up to now, but you need to find the cell culture medium that is good for all cell types.
Yeah, and this is pretty hard to find.
It is.
Okay, coming to your questions. The first is, how do you tackle process variability of 3D bioprinting, where we are seeing issues where bioprinters print differently depending on the day and the operator? I think this is a question to you.
Actually, this is what I wanted to show, that using an automated mixing procedure, for example, drastically reduces variability. But obviously, we think that this is a major issue, and that's just on training.
Okay. And do you see hyperproliferative, behavior of cancer organoids as one would expect from human cancer?
Openly said, we have not checked that. I mean, the spheroids grow, so they are proliferative. But of course, we just developed the model, and now we have to analyze it in more detail on how far it really reflects the biological situation.
Okay, and the next question is to you both. What is the physiologic explanation of the fat flap growth filling the lattice cage?
Okay, the implantation of Lattice induces a transient acute inflammatory response. As you can this literal layer of connective tissue contains inflammatory cells that produce angiogenic and adipogenic factors that diffuse through the implant, and they feed the fat flap. That's the explanation. And you have an increase in the number of cells. It's really a cell proliferation, so stem cell proliferation, then differentiation in pre-adipocytes, and after in mature adipocytes. And it's not an increase in fat volume. So this really an increase in the cell number and not the cell volume. And it's very important because the increase in cell volume depends on the nutrition, on the diet, and so it can be regulated with obesity or fasting. It's not the case here because it's only the increase in cell number.
Mm-hmm. And I think the next question also addresses to you. What do you mean when you say your filaments are medical grade? This is one of my favorite questions also.
Okay. So it's based on medical grade pellets. So for instance, it's a GMP production batch of pellets. And it's called medical grade since the toxicity is compliant to the ISO 10993.
Okay, that's it. I think the next question is very similar.
You have a file. You have a file on the GMP product itself. Yeah.
Okay. So next question: are the best implants especially treated or filled to influence the ingrowth of cells and acceptance of the body?
Yeah, there is no chemical, what we call chemical or surface treatment. So it's a pure biomaterial itself. One thing Philippe did not say also in the influence of growth, it's also a mechanical induce. Mm-hmm. You have some kind of mechanical production of the tissue, and so the tissue also expands in the space, and when it reaches the edges of the implant, it stops. So this is how we can adjust the volume of the breast. So no, there is no treatment, only what we call a post-processing is to remove the support material of the printing.
Mm-hmm. Okay, because the cells, how do you think, do the cells know when they should stop with growing? Because these are not cancer cells, they are fat cells, they don't grow forever, they have to stop, and so they know this?
It stops, it's a contact inhibition, so it's like when you broke a bone, you know, the bone cells are conducting and repair, and it stops when it reaches the edges, and it's like yes. The same here.
Mm-hmm. It stops with the compression of surrounding tissue.
Okay. Okay. What do you mean by, we have this medical grade filaments. How is the shape stability over time? This is also a question for you.
So at clinical level, we don't have enough data to answer yet, but at preclinical level, you've seen the results on the kinetic curve. So once Philippe said that it's angiogenesis, so we have a regeneration. We have a number of cells increasing over time. So we have some kind of new tissue, and this new tissue is stable over 21 months of observation. So after the full degradation of the biopolymer, the tissue stays stable on volume. So, and we can confirm this because it's vascularized. You've seen the technical operation. We transfer the flap from here to here and still vascularized, so we can ensure that the vascularization is still yet. And so we expect the same results on human, and let's say next year,
okay.
After 1 year review.
And we have another question for you. Your bioprinted models are devoid of animal components. Are you not using fetal bovine serum and other animal-sourced materials in culturing as well?
Thank you for this perfect question. I might have typed it myself because this is the main effort in my lab right now. I didn't have time to go into this. Just two short remarks for those who are not too deep into this field. First of all, everybody who uses cell culture uses fetal calf serum, which is obtained from the fetus of a slaughtered cow. And secondly, most people doing bioprinting or using organoid technology or whatever use basement membrane extract, which is known as matrigel. That is even harvested from implanted sarcoma into mice. And that's what everybody does. And we recently did a systematic review on bioprinted liver models. All models contain these components and are sold as animal replacement methods. With the models I've shown you, we also included these substances from animals. But we have a manuscript currently under revision, which we think is the first xeno-free liver model produced by bioprinting. So we adapted the liver cells to FCS-free conditions in chemically defined media. And we replaced all animal components in the bioinks. And we think that this is the future, not only to protect the animals, but also to have a pure human system, because it doesn't make sense to combine human cells and then have them in a fetal serum again. And coming back to your question, it even makes things more complicated if you have multiple cell types. But recently there was a publication from a Swedish group, which we now want to follow on, that has what they claim a universal FCS-free media, which can be used for any cell type. So this is really the future of these bioprinted organ models. Thank you for this question.
And I would like to add another question, because Berlin has declared its goal for becoming the first animal-free capital. So what do you think? How far away are we from achieving this, and when will we succeed?
I don't think that this is realistic, at least not in the foreseeable future. Of course, there's an ongoing discussion, but we have the Charité here, we have a lot of research networks here, so we will not be an animal-free city in the near future. What we can do is to provide good models that may be used as replacement and offer them to the biomedical research community. That's all I want to say.
It's clear.
It's too dangerous, the politics behind.
Yeah, but it's clear. Next question. You rely on the toxicity treating in the material only, or did you test according to 10993 on the final device and production process? I think this is a question for you.
For instance, for Lattice and other device, we've done the biocompatibility on the produced device on the production platform you've seen. For the filament, if I try to understand the questions, it's done on the raw material itself, but if you want to develop a CE mark of FDA cleared device, you have to make the test on the final device.
Okay, thanks. And next question to you, Julien. What is the mechanism of action of your device?
I think Philippe has already answered this question.
Philippe?
So there are two different mechanisms. One is mechanical, because the implant creates a protected space to allow the growth of the fat flap without the compression of surrounding tissue. And the second is a biochemical, because it induces an acute and transit inflammatory response, which mimics a wound healing process. And there's a local production of angiogenic and adipogenic factors that fill the fat flap and allow the growing.
Okay. And do you have other devices in development? What are your next steps?
Yeah, so next steps for Lattice are the end of clinical trial and CE mark expected in 2026. We have started the discussions with FDA and plan for a breakthrough device and PMA process. So it will be a really long road and really expensive for US. And we have the second device, Rodin, for the skin regeneration after deep wound or burns, and Noelis for meshes in soft tissue.
Okay. And are you referring to channels when you say vascularization? Because vascularity is biologically different. Is the next question or comment.
Okay, yeah, we use the term vascularization. We have the channels, but what we're currently working on is to add growth factors so that there is neovascularization from these initially printed channels to have microvessels that go deeper into the tissue.
Okay. Next question, I think, is still answered. It's a question concerning the stop of cell growing, and you told us that the cells stop if they reach the edge of the cage.
The edge of implant, it stops, or the skin, if the implant is over, the compression of the skin of the implant will stop the growing.
Okay, then we have another question. Have you seen any difference in the shape after sterilization? This means the cage without cells?
No. We have some kind of specific treatment, thermal treatment during the process to ensure this will not change. And the sterilization is ethylene oxide, so it has a less impact on the biomaterial itself.
Okay, and I think we have time for 2 more questions. Can the dissolvable implant material also be used for other implants? And it could be interesting to use it for fixation of bone fractures, perhaps?
Yeah, of course, it's common range of polymers already used in medical applications for meshes, for cranial implant, and so on. The use of 3D printing is really interesting to make tissue engineering, and bones is a real application. I have not mentioned it, but in our broad range of machine, we can do twin screw extrusion to add. It's an echo for the 3D systems, and the presentation of this morning, they use drugs into the polymer, so we can also do the same thing to add, for example, hydroxyapatite into the PLGA matrix to ensure diffusion of and regeneration of osteoblasts into the bones. Yeah, so this is totally possible.
Mm-hmm. And would you say 4 to 6 months post-unilateral mastectomy, the breasts are close to systematical appearing?
It's not easy to say. Real symmetrically, it's difficult. It's really difficult. So maybe depending on the case, if the volume is almost identical, you can reach this symmetry. If not, the patient will have what we call a lipofilling. It's an ambulatory surgery to inject fat to make the symmetrization.
Mm-hmm. And maybe last question, do you see any issue with seroma in the dead volume of the wound? Not yet. No, not yet. Not yet. Okay, one very, very, very last question. What have the results of pre-testing shown?
Of pre-testing?
The preclinical tests?
The preclinical tests. So, preclinical tests, regarding regulations, CE or FDA, finally, you have to prove 2 things. The safety of your product, so you have the whole guidelines of biocompatibility to ensure that your process is safe and your biomaterials induced by your process is safe. And then you have to prove the performance. So the performance shown on the slides here is the regeneration of tissue. So this is one criteria of performance. And the stability of volume after degradation.
Okay. So thanks a lot again. All the other questions could be answered during the next break, for example.
I see a question about radiation. We have published an article with our implant and radiation. So
okay.
It works.
And you can discuss this during the lunch break, for example. So thanks again for the fantastic presentations.